Well, good morning. You guys have already heard a little bit about the UCR collection from two of yesterday's talks, so I'm trying not to go over the same ground. In fact, I want to focus a little bit more about, as the title here says, what's in your collections and not just so much what's in ours, because I think we all have a common issue. And I want to address the common issue a little bit more and use our collection as an example. Which is the advance? Should be good. Okay. Get my notes. Every insect collection is in some ways idiosyncratic. Every collection reflects decades to centuries of accumulated contributions from many different and unique sources. So we have, a variety of different places from which we acquire material. And every collection has in it somewhere something that is not from where you would expect. So it might be geographically anomalous. It might be not the sort of taxon you would expect to find in that collection. So it would be taxonomically anomalous, or perhaps simply not on the scale you would expect as well. And often this is the historical legacy of some person or people who may or may not have been known well outside of your institution. In some cases, even someone completely unconnected to your institution. So it could be an historically exceptional thing that somebody wouldn't think, oh, that's going to be in this person's collection. There's a known history for every collection, and then there is the unknown history. And so that has to be something that you might know about, but might need to be a little bit more widely shared. So what this means is that you might have hidden treasures. We all have hidden treasures, things that just lurk where no one would expect to find them, like that. Oh, Frodo. So if people don't suspect that you have something, what are you going to do about it? These people unaffiliated with the collection, containing treasures largely hidden to the outside world, even when it may be extremely valuable. The bigger the collection, the more likely this is to be a problem, especially in terms of its scope, the numbers of specimens, the diversity of taxa, and in terms of the impact on the use of that collection. A small collection is not likely to harbor as many treasures, but it's still something to keep in mind. We don't manage insect collections for ourselves. This is a fundamental thing. We are part of a community, and it's essentially shared property, and it's important to be able to assess what that shared property is. We manage the collections for the benefit of the global community, and that benefit is greatly reduced if there are hidden resources in your collection that need broader exposure. Really, that's the primary thing that I want to drive home here today. We all should think about what sorts of hidden treasures respective collections contain and what we can do to bring them to light. In that vein, using UCR's collection as an example, our collection reflects more than anything else the contributions of this particular person. Philip Hunter Timberlake. It's not Paul, it's Philip. His wife was Richard Nixon's uncle, so he's one of the few entomologists who we know was a personal invitee to a presidential inauguration. When Nixon became president, he attended the ceremony along with his wife, who had put Richard Nixon through law school. So he has a little of a historical connection outside of entomology. He began organizing the nucleus of the UCR collection before there was a UCR collection, before there was a UCR itself. So this was back when it was just the Citrus Experiment Station over a century ago. And out of the 4 million specimens in our collection, he personally collected over 500,000. He collected them by hand. He didn't use traps. He labeled each specimen by hand. And every specimen has like 4 handwritten labels on it. He cut his points by hand. Everything he did meticulously by hand, and he had time to do all of that. I mean, that on the upper left, that's a pretty typical unit tray of Timberlake specimens. They're packed in like sardines. You know, and they're almost illegible. There's only a handful of people around who can read his labels, but it's incredibly invaluable material, especially because he was one of the first people to make really extensive notes on every single specimen that he collected as to what the host plant was that he was finding on. And this was when it was not really a common practice back in the 1920s and 30s. It wasn't exactly the standard thing to see the flowers identified the species that people were collecting bees on. So he generated an incredible amount of information and an incredible number of specimens. But even though he's known for bees, he did other things. Um, he yeah, he brought thousands of specimens with him. He had been working in Hawaii. He brought all these things that are endemic Hawaiian taxa, the Actemnius, Odinoris, Protorhinus, and numerous carabids. Jim Liebherr just recently did a revision, and Timberlake's carabid collection from Hawaii was an essential part of that revision. And because Jim knew that we had the material, he knew that he could borrow it, and it was a win-win situation. A lot of those insects are probably now extinct. And because these were collected in 1910 through 1920, a very long time ago. And on top of that, he had left behind all this stuff when he died that he had never gotten around to working on, but he had put manuscript species names on hundreds and hundreds of species. We just had Joel Gardner come through a while ago, look at the Dialectus. We had 150 species that Timberlake had set aside as new, and it turned out that 50 of them were synonyms. 50 of them were things that somebody else described since Timberlake figured out that it was new. So they were all new species, but they had been given names in the intervening years. And 50 of them were still things that nobody had ever seen before and were still new taxa. So Timberlake also had a really good eye for when something was new or not. Now, once UCR actually came into existence, we had contributions from a number of important faculty researchers. And I just put one up who was mentioned yesterday as an example. He's been retired for a while now, but he continued to contribute to our collection long after his retirement. And he's an especially interesting example because he worked on lots of different things, and every single thing he worked on, he really enriched the collection and gave us, a world-class collection of whatever taxon that it was that he worked on. He worked on trichogrammatids, he worked on meloids, and he worked on mirids. So he was in 3 completely different orders. And every one of them, he went in, gung-ho, built up a collection, built up the synoptic material, and gave us, a rich legacy of stuff. If one person knows, oh yeah, I work on trichogrammatids, the Pinto collections at UCR, they might not know anything about the meloids or the mirids, and vice versa. For people who know mirids, they wouldn't know that he left a collection of meloids behind, and so forth. So the legacy of an individual professor may be something that is also not completely appreciated by people outside of the institution who is the host. There are a number of other past faculty members who've contributed, such as George Giorgio, who deposited many agricultural pests and other insects from Cyprus, Dick Gaydon, who worked on tephritid flies, and there's still active faculty members like Christian Weirauk, working on mirids and rediviids, John Harity, who works on chalcidoid wasps, and their numerous students, plus other faculty like Mark Hoddle, who's contributed massive numbers of Malays trap samples from New Zealand and Guatemala, which is a collateral benefit of his fieldwork on invasive species. So our faculty have been helping build the collection as part of their work, but they're not the only people who contribute. There's also a large amount of material that comes in from non-faculty and volunteer contributions. Curators, non-faculty staff, students, volunteers, and outside donations account for much of the really noteworthy material. And this brief summary today doesn't really do it justice. Yes, this group includes myself, my boss, Sergey Tryapitsin, the former collection manager, Saul Frommer, and also people who volunteer or have peripheral associates like Adrian Mayer, Greg Ballmer, Mike Irwin, Jack Hall, Gordon Pratt, Dave Hawkes, Noel McFarland, Frank Pelsu, and others, each of whose contributions numbers in the thousands to hundreds of thousands, or tens of thousands. The taxonomic and geographic limits go far beyond what people might expect for a Southern California collection, and we're proud to serve as a repository for all of this material from all these different sources. With this much stuff to share, it's not always easy for us or any large collection to put all of that material to good use, and it's not always easy to get the word out about what you have. How do we do that? Well, there's many ways. We have a website, and it tells a lot of the same story I've already told, but people have to seek that out to find it and get that information. We try to interact via social media platforms, but that can be a big time drain and might not be easy to maintain. Our Facebook page was hacked, for example. We had one for a number of years, and it was destroyed. And there are no fewer than 3 fake Facebook pages claiming to be ours that aren't, and Facebook won't get rid of them. It's a nightmare. So that has not worked out too well. Then we have meeting talks like this one, and that's another good way to spread the word. I've been coming to ECN meetings now for more than 20 years, and I've heard a lot of talks describing various collections, giving their history, but surprisingly few of these talks have actually, talk about what's in the collection that you don't expect to see there. And I think that's a wasted opportunity, to be honest. If experts are unlikely to find your collections, then maybe you can invest a little time and energy in finding the experts. If you've just acquired, say, a pile of grasshoppers from Sri Lanka, maybe you can do a little digging into who the experts are, find out if there's anyone actively doing research on that group, and contact that person yourself. It's a bit more legwork, but that can pay very big dividends if you find the right person. Personally, however, I think that what is ultimately the most important thing any of us can do is to share our data online, because that will show up when people are looking. And it could involve central places to distribute that information, such as the ECN website. That's something to consider. People from the taxonomists to ecologists do or should make use of online data portals like GBIF, iDigBio, Scan, Discover Life, and all those things. And this is a very good way to let everyone know and see what sort of treasures you've got hidden away in your collections. If you're databasing your collection, however, consider prioritizing that sort of material. Seriously. The parts of your collection that you don't think people are going to expect to find there might be the most valuable. So consider skipping the obvious stuff and focusing on the obscure material. If you're, say, a collection in Michigan, but you have, suppose you have a nice collection of, I don't know, Costa Rican odonates that just happen to be there, then think about databasing that material first, rather than trying to just work on the material from Michigan, because everyone's going to know that you've got a load of stuff from Michigan, but they're not going to know that you have those Costa Rican odonates, are they? No one is ever likely to make use of them unless they know that they're there, and that would be a shame. If your collection is has got a particular taxon that was meticulously curated and ID'd by an expert who isn't necessarily known to be an associate of your collection, a global authority, consider prioritizing that material as well. That's the most reliably identified stuff, and it's historically valuable because it was worked on by an expert. The expert IDs are especially valuable now with the paring down of the taxonomic expertise in the community, and we're being usurped by AI tools. Those are never going to be able to compete with real taxonomists. So embrace the things that are special, different, and unique in your collections and draw attention to them. If you do that, everybody wins. Thank you.
We have maybe 1 or 2 short keys.
It's maybe focus here to have a symposium on the treasures, send out questionnaire to the whole ECN so everybody contributes.
Thanks, I like the last slide. Can you tell us how much you actually also put identifiers on people names? I just wanted to say that's also a really great way to discover who else has collections from the same person. So it's what gives you unique, but it's also what helps you find out who else has already teased out that challenging campaign, right? So at the same time that you get your database of specimens, they have identifiers on people. You do that work too, yes.
We do have, our database has a field for the collectors. It's not the highest priority field to fill when we're doing stuff that has to be data, legacy material. If it's a person whose name we don't recognize, we're generally not taking the extra time to put that in because if it's not a familiar name, it's not likely to be, anything that's going to be helpful to people. And, we have very limited manpower, 1 person working in a collection of 4 million specimens. And a lot of the databasing and stuff I've done myself. So got to prioritize.
This is a pitch, and for me there to figure out how to capitalize this because it's another way to do everything. It's another way to discover our uniqueness, and it's another way to discover somebody's identity.
Right.
And that's easy.
Our next speaker is Brennan Hayes from ASU.
Before I get started, I just wanted to say I think we should do a little round of applause for everybody who's in charge of keeping the PowerPoint slides going and the tag and and that's in no way because Andrew's a personal friend who texted me at 8:05 saying, "Why isn't your slides uploaded?" Well, anyway, hi everyone. My name is Brennan Hayes. I'm a master's student here at ASU in the Nico Franz lab. And today I'm going to be talking to you about EcoPalette, which is sort of just an attempt and a foray and an exploration into potentially trying to figure out how we can visually encode environmental data characteristics into images themselves for the visual machine learning process for species identification. And so this talk is going to be about the model in part, but also a lot of the talk, because it's in its kind of infancy stages, is going to be about the ideation phase and the workflow, because as I think a lot of people begin exploring this, I think it's good to start sharing some of that at the beginning. And so just to get into it, I want to talk a little bit about the inspiration and maybe uniquely or weirdly for where did this all start? Well, it kind of starts with this movie called Arrival. Which if you don't know what Arrival's about, it's basically where an alien race of squid-like aliens comes and tries to communicate with humanity. And humans, in coming to decipher their language, and I apologize if I'm spoiling the movie for you, they come to discover that the squid-like aliens' inky circular language is spatio-temporally contextual. So it contains both past and present and future and space all within that language. And it kind of unlocks humanity's way of thinking about themselves in the future. And I was just kind of enamored with this idea, right? This idea that the way that you talk about something, the way that you visualize language changes our understanding of it. And so I kind of began wondering, what context do we assert on the individual specimen? And perhaps like most of you in the room, you're likely to kind of go to, well, the extended specimen concept, right? That's kind of the context. that we place around a specimen. You take a specimen out of its environment, and then you have that physical specimen, and then anything that comes off of that, any of the extraneous data, like images or the digital specimen record, are inextricably linked back to that physical specimen. And I think that this is kind of a really great answer, but to me, as kind of an ecologist, it always felt mildly unsatisfactory in this small way, and I couldn't quite put a pin on it until I verbalized it as, really the specimen is an extension of its environment. That's what the specimen is. It's a moment in time, right? It's the spatiotemporal context of the environment that it was in. And so as I entered into my master's thesis, which was working with NEON, National Ecological Observatory Network data, I sort of kept mulling over this idea of how do we contextualize specimens. And that led to a point where I was giving a talk at iDig Bio on NEON data. And while I was there, Eric Sokol from NEON came up and he was talking to me about my talk and then invited me to attend Beetlepalooza. And I said, what the heck is Beetlepalooza? And he explained that Beetlepalooza was this attempt hosted by the Imagomics Institute at OSU and NEON to bring together a bunch of different researchers from various disciplines to work on this kind of advent of visual machine learning. And we have all these NEON specimens, we have all these images. What do we do with these? And I was intrigued, very humbled that he asked. And so I naturally asked, I think the question that I hope, somebody like my advisor would be proud of me for saying, which was, y'all got any money? And the response that I got was, yeah. And this is a rough approximation of our conversation. And so I attended, and I was worried going in that I was going to be kind of the black sheep, and I didn't know where I was going to fit, but I was really happy with the makeup of the attendees. So there were about 30-ish people there, and we had about 16 half people who were machine learning experts, about 10 ecologists, and about 4 systematists. I'm sure everyone in this room wishes that number was a little bit bigger, but I think it's a pretty good spread. And we were able to pitch some of our ideas, right, on that first day. And a lot of the ecologists kind of went for, what I thought was sort of the rote answer, which was, let's take the systematist work and their knowledge, let's plug it into some cool fancy model that identifies the species, and then let's plug that into an ecology model. And maybe coming from a systematics lab, I didn't love that answer. And so I proposed that we do the opposite. Let's instead try to contextualize the individual specimen by asserting the ecological variables that would have been at that time and space into the image itself. And I was lucky enough to get a team of 4 other people and myself who were kind of taken by that idea. And so let's kind of dive into the project now. We worked with NEON sites. If you're not familiar, NEON, it's a big ecological monitoring project, spans the US. We specifically looked at these sites sort of to begin with, because they provided us with a kind of limited data set to operate with, in part because images was what we were working with. So this is these images were taken by Isadora Fluck out of Florida. She went in and took these falcon tubes, which are pooled specimens from multiple collecting traps at the same time, and laid them all out and took all of these images. And a big problem in visual machine learning, if you don't know, is called segmentation. So it's trying to get the individual specimen out of maybe an image of many specimens. And we had one team who worked exclusively on that, which was really useful, and we all benefited from that. And then I was able to kind of adapt some of my master's thesis work, which is part of the R code. That's why I've hidden a little bit of it, so that we could take NEON environmental data and inject it into the image. And we were trying to figure out, how do we want this to look? What did we want this to be? And we kind of came up with this rough idea. So we thought about, having the image in the center and then chips of color that would be representative of that information of the time and space, right? The place, the physical location, the rainfall, the temperature, normalizing that, and then using hue and coloration, saturation as sort of the change in that. And so we did that. So we plugged in the NEON environmental data. We had our cropped images that were segmented, and then we threw that in the palette script. And we got some chipped images on the outset. And so this is kind of roughly what it looks like. And so this is the image that we were then wanting to feed into the visual machine learning, into the AI model. Now, I do want to be very clear here. We are not and have not used all of these chips because I don't know if you know this, but data wrangling is hard. And so this is kind of more like what we're looking at right now, but we're actively exploring and adding in new pieces of information to try to see how the model handles it. And this was sort of the rough makeup that we were working with for this first iteration. You'll notice here, it's not a very, very large taxonomic. There's not a lot going on here, but again, limited, right? It's all in scope. We threw it into some RoboFlow training. So this is kind of an initial foray into it. And we found that without eco-palette validation, we had about 88.1% accuracy. Again, very small subset. And then with eco-palette, we had 99.7. And hopefully everyone in this room looks at that 99.7 and goes, well, that's not right. There's no way. And you're very right. It's very overfit. And that's kind of one of the active problems and something that we wanted to share about the workflow is, yeah, overfitting is a huge issue here. And so we're also working with BioClip, which is also done by Imagenomics. It's kind of based off of OpenAI's Clip, which is all about kind of creating a generalized model that can identify species. It's really great and it's open. I believe it's open source and the team is really wonderful. And I highly recommend it as an access, as a resource. So we fed all these images, we kind of got out some results and we had this question of, well, what is the AI looking at? And it turns out it can be kind of funky to figure out what the model's looking at. And so we used this way of kind of creating a boundary box to look at it. And we found, hopefully you'll notice right away, yeah, it goes straight for, in every image, it was going straight for the chips, right? It wanted that environmental data. It was cheating a lot of the time, right? But still, it was using the environmental data, right? That the chips that we were inserting was interesting to the model and it was wanting to use it to kind of help create its delineations. You'll notice, however, hopefully that also, it also ignores some characters that are probably important. So we have like some genitalia peeking out here that it's like, I don't care about that. I want to see what the funding colors do. And so that's kind of one of the things that we're wrestling with at the moment. We know that there are some potential pitfalls here. Oh, sorry, I have a joke. There you go. We know that we have some potential pitfalls here. Diversity of training data, overfitting. What are the goals that we're trying to figure out? On my side with the environmental stuff, it's missingness a lot of the time. The neon data has issues, but all environmental data has issues. And how do you handle that and defining that? And then for the images, what quality and size and how is the specimen orientated and all of that is something that we're having to actively work with because it's a very iterative process. So what's next? What are the things that we're trying to do now? One of the big things is we're trying to get a better way of looking at the model is doing. So we want to implement a thing called GradCam or at least a variation of it, which gives a more detailed heat map that shows much more specifically down to the pixel where the AI is roughly looking. We're also working on adding more data in and seeing how the model can handle this. So this is an example where it's hard to see, but on the far right, just this week, we were able to get in a neon NLCD class. So we're looking at vegetation, we're looking at percent coverage, incorporating that. We're looking at soil composition and incorporating that into the model. On my side, I'm big on interoperability and I think, neon's great, but I would like to be able to use other data sets. And so I looked at NOAA's GHCN data set, which is a 6,000 stations kind of across the US. And so I've been looking at that. The problem is it's pretty big. It's about 75 million rows. So my computer doesn't love that. But that's kind of one of the goals here is, right? You use a weather station nearby that is relevant to you or use a conglomeration of weather stations to generate some of this data to feed in. So to end on, I kind of wanted to address maybe a little bit of the elephant in the room, which is, will machine learning or AI replace the role of systematists? And I don't think so. I really don't. And the example I use is the Swiss cheese model, which if you're not familiar with this is from Lab Muffin Science Beauty. she's using it to explain skin cancer and how to prevent it, and how, you put on sunscreen, but you don't put it on perfectly. It's got some issues. And so you wear a hat and you stand in the shade, right? And each one of these things is imperfect on its own. But by combining each of these fallible slices, you create a more infallible whole. And that's where I think we're going with here. The purpose here is not, and should not be, to create some omnipotent AI deity. It's rather to develop a relationship with AI built on trust and an understanding of where each excels and the other falters. It's identifying the holes in the Swiss cheese and building processes and infrastructure that's both physical and digital to facilitate coverage, which makes each fallible layer that infallible whole. So to end, I just also want to say thank you very much to everybody who has been instrumental in this project. That's my contact information. I'm in the PhD market. There's a org ID, and yeah, I'm happy to take any questions at this time. Okay.
Make that our inspiration, sort of the platform of this prosecutors. I just want to ask some data concepts there.
Yeah, so this is actually from Isadora Fluck out of Florida. She was looking at morphological change in characteristics and trying to figure out if there was a way to sort of segment the image and annotate them to sort of view that. And I think a sort of sub question I have there is, well, I won't get into that, but this was her idea because she used glass, and glass was reflective. It did not work very well. And so she tried out a couple options, and these are non-pin specimen, they're pooled. So this was sort of what she ended up with was here, and it worked well for our purposes. We didn't find major issues. Yeah, exactly. Yeah, that was her explanation, was that, it could tip really easily. And I apologize. The question was asking why we use Q-tips for the Zoom audience.
Our next speaker is Max Barclay from the NHM.
Excellent. Thank you very much. So I'm afraid that my talk is going to be like many of my talks, broadly philosophical, and not so much talking about what I've been doing, but why we're all doing what we're doing. So I called it Develop or Die, why collections must remain active to remain relevant. And I put the abstract in the program. But basically, I'm talking about irrelevance as an agent of deterioration. Entomological collection management is an ongoing trade-off between protection and exposure of the material. On the one hand, we want to pass as comprehensive an archive of the natural world as possible onto the next generation. On the other hand, we have to expend our time and resources to increase scientific access, because without exposure to the scientific community, collections stagnate, and all that we would have left to pass on to posterity is a legacy collection, a historical anachronism, a museum of a museum, and that's obviously what we want to avoid becoming. Now, there are obvious exceptions to this. Examples like the Linnaean collection is the collection of Linnaeus. Linnaeus is dead. That would be stored in its original housing under its original combinations and provided access so that people can confirm their identifications based on Linnaeus. So that's a legacy collection and is by definition a legacy collection. And there'd be no point going out and collecting more material and adding it to that. But as a curator, I think one needs to have an eye constantly on the past, another eye on the present, and another eye on the future. So you have to have three eyes. And you shouldn't be concerned or worried about the past, the present, or the future. You need to keep an equal footing in all three of these. We all have to deal in museums with agents of deterioration. This is the MNH's list of agents of deterioration. Things like light, temperature, pollutants, pests, physical forces, theft, fire, water, etc. And that's obviously all very important and part of our day-to-day work. But what we neglect is that we can protect our collections from all of these and keep our collections entirely safe, but we're still subject, if we're not careful, to the slow, insidious, creeping decay of irrelevance, which we may not even notice if the collection is in good condition. The sort of dead collection walking is what I'm very concerned about becoming. And some major European collections, what I love to do is come to the ESA and the ECN and meet all you guys and see all of the activity and all of the dynamism that's going on and all of the things that you guys are doing with your collections. Because you have major museums in Europe, maybe the most important museum in Europe, that are more or less legacy collections now and that are not making their types available, they're not making their materials available. And it is a serious obstacle to science, and it's something that we could all become if we were not careful. There's a quotation here from Joseph Grinnell, who is the director of vertebrate zoology at the University of California. A museum's policy should be everywhere as liberal regards the loaning of material to non-resident as well as nearby specialists. The value of a museum's hoard of specimens and facts increases in direct ratio to the extent in which they are used. No museum is a success as long as it remains a cold storage warehouse. That's more than 100 years ago. This guy is a fish curator, but the similarities to what I believe and what I think we should be doing are extremely apparent. But if we protect our collections from all of these problems and keep them entirely safe, we're still subject to this slow decay. Now, I gave a talk last year in, 2 years ago, I think, in Vancouver about insect collections in an age of increasing relevance and decreasing recognition. I was talking about the quotation from the Lord of the Rings. I titled it as Fighting the Long Defeat. This is a quotation from Galadriel. Once that much was is lost, but none who live now remember it. Together through the ages of the world, we have fought the long defeat. I was probably being a little bit morose about 2 years ago. The reason I was being morose is because at that time we had just acquired a major 19th century Lepidoptera collection from a private school. And at the same time, we'd acquired a major collection of Cerambycids from a regional museum in the north of England, a collection of Elphinstone Forest Gilmore. And these are two major legacy collections that have come to the Natural History Museum. And my concern is that while initially this was exciting to have obtained these fantastic collections and made them available for research, there was also a concern that children are taught about habitat destruction, they're taught about climate change, they're taught about extinction, they're taught about the insect apocalypse. And here we have an exclusive private school and a local museum who are in the unique position of having inherited remarkable tools to answer interesting questions about these phenomena. And the inclination of these organizations is to get rid of those tools. And that made me very morose, even though in many ways we were the beneficiary of this because we were the institution that was going to make these things available. On the other hand, it would be lovely to think that there were schools and museums all over the country and all over the world that were making collections available to their audiences. But in retrospect, and 2 years later, we've made serious progress with this Gilmore collection of Cerambycids. And we've got 4 papers in press with new species described, with catalogs of his types, with photographs of things that have never been photographed and made available before. And that collection has effectively been sitting in an attic for 60 years. And much as I might want to believe that it was accessible and that it would be good to have hubs of accessibility all over the place, it wasn't. It was a closed legacy collection, and now it's an open and available collection. So perhaps it's a good thing. I really have mixed feelings about this. Private collections, I have often said, are like streams and tributaries that eventually flow into the larger rivers of the big institutional collections, and that ultimately feeds into the sea of human knowledge. And with the destruction of natural habitats, the urgency to collect and document biodiversity continuously grows, and the relevance of natural history collections will only increase. Quotation from my colleague Richard Fortey: collections are the conscience of the world. They show us not only what we have, but also what we've lost. This is in a meeting of the Royal Geographical Society. Another quote from a great American, Quentin Wheeler: Why is taxonomy important today? He said this was in the early 90s. I think future generations will be appalled with us that it was so easy to explore biodiversity and we chose not to. And then he added, rather controversially perhaps, because we were doing parlor tricks with DNA at the time, that everything was going extinct. It is slightly controversial, but 30 years ago in 1990s, Quentin perhaps saw DNA as something that was going to replace taxonomy, just as the last talk was talking about AI, and that there were some concerns amongst some people that AI is going to replace taxonomists, or that digitization is going to replace taxonomists. We all see these threats, and these things, I believe, are not threats. In the 1990s, they thought DNA was the enemy. Now we have integrated DNA into the toolkits of collections and major museums. I personally don't believe that DNA threatens to replace curators and scientists. I don't believe that digitization is a threat either. I think we should embrace all of these techniques. And we've embraced digitization in the Coleoptera collection, which I manage at the Natural History Museum. We're striving to get the whole collection, 27,000 drawers, available online. There are already benefits from this. We're about a third of the way through it now. Curators have more control and awareness of loss and damage. It facilitates offsite or remote working. It speeds all curation tasks. It raises the profile of the collection internationally and facilitates collaboration. I hope there's people in this room who have ported into our data portal and looked through our collections without having to travel to London and find accommodation and get on an airplane or anything like that. And also, many inquiries become self-resolved. A lot of people come in and inquire and say, what color is this species? Or do you have any specimens of that? Or do you have the type of that? And now I'm hoping people can just port in and see that we've got that and we've got this. And so it's in some ways a labor saver. But to me, the greatest existential threat to what we do is collections becoming the walking dead, becoming an irrelevance, beautifully preserved, fresh looking, but devoid of life and interaction and activity. So I want to introduce the concept of Schrödinger's curator. Now, we all know the paradox of Schrödinger's cat, that the cat is simultaneously dead and alive in the box, that it may be dead and it may be alive, and you have no way of telling because you haven't opened the box. Schrödinger's curator, to me, he believes, or she believes, specimens are so valuable that they shouldn't be sent out on loan, they shouldn't be handled by digitizers, they shouldn't be used for exhibition, they shouldn't be handled by visiting scientists. We've got really precious stuff here, but they're so worthless that we don't need to collect anymore. We don't need to invest in the infrastructure. We don't need to acquire new collections. We don't need to upgrade or maintain our identifications. The Schrödinger's paradox that collections at the same time are incredibly valuable and should be preserved, and at the same time are worthless and not worth investing in and not worth developing and not worth growing. And Schrödinger's curator, or maybe Schrödinger's administrator, some of us might say, has taken control of quite a lot of institutions and is a figure that we should be aware of and we should have a name for, because these incompatible views seem to be held simultaneously by some people. The result is that Schrödinger's museums just sit on their collections like a dragon on a pile of gold, not making them available. We are at the Coleoptera section in London. We're making a lot of material available by loan. We're getting a lot of material identified by specialists all over the world. And the collection is growing by 500 to 600 type specimens per year that are identified from unidentified material because we're sending it out on loan. Now, if we said the types are so precious, we're not going to send them out on loan, then we would be perhaps saving some of our types that we already have from risk. But the cost would be those hundreds of type specimens that are being generated by the loan system. So the net loss would be hundreds of types a year. So in order to protect maybe one or two types, like some famous European museums might have decided, we are losing the potential to grow science and scientific knowledge by hundreds of species a year that are being described from our material. Also, if you're sitting on your collection like a dragon on a pile of gold, your threat is not digitization, it's not DNA, it's not AI, it's not any of those things. It's somebody saying, we can replace you with a good climate-controlled storage and a good lock. And that is all that we need to protect this collection into the future. So we have to remember what Joseph Grinnell said more than 100 years ago. And we have to remember that we have to remain relevant and we have to keep the collections open. I'll just finish on two quotations from great Americans. Those of us who would give up liberty to purchase a little safety deserve neither liberty nor safety, attributed to Benjamin Franklin. And another quotation that I've modified from one of your presidents. I'm sure that presidents are a little bit of a sore subject for some people at the moment, but do not ask what your collection can do for you. Ask what you can do for your collection. Thank you. Okay.
That was very sad because I was also morose and I was looking at your definitions of Schrodinger's collection. Is that the administrator's view of Schrodinger's taxonomists? And what leads to the slow erosion of those people that were borrowing those specimens in the 80s.
You think that the population of taxonomists who can identify things is declining because of that syndrome as well? Is that what you're saying? I think that's inevitably true, and that's why we have to hold the torch and we have to look to the next generation of taxonomists. And I think that museums being open and available to younger people is actually the key to ensuring a next generation of taxonomists. And I remember when I was the youngest person in this room, and I'm not the youngest person in this room now by any stretch of the imagination. So there is some progress going on, perhaps, but I think that we all have to remember that public outreach is not something that we do for vanity. It's not something we do for the museum's reputation. It's something we do because we want to catch the one person in 100 who's going to become the next expert on Bombylidae or something like that. And even if they're going to do something irrelevant like Diptera or Hymenoptera, it's still, it's still catching people and bringing them into the fold. And I think that's what we need to do. Excellent.
And our next speaker is Jennifer Girón from the Museum of Texas Tech University.
Good morning, everyone. Again, this is me, Jennifer Girón from Texas Tech University. And today I'm going to talk about a particular project that has been bringing specimens into our collection. And it's a collaborative project on the Davis Mountains Preserve, which is a really cool place in Texas. And the emphasis of this talk is going to be not on the methods that we are using for collecting the specimens or the logistical things that go on in the field, but more what are we doing with the specimens once they get to the collection. This is a project with a lot of people from a lot of institutions who go out in the field every year since 2021 to bring specimens for documenting the arthropod diversity of the preserve. So for those of you who are not familiar with Texas, and I'm not familiar with it myself, it's mostly plains. In green, you can see the coastal plains. In yellow, the north central plains, and in orange, the Great Plains. So not a lot of mountains in Texas, except for this corner in pink, which is the mountains and basins of the state. So in this region, we have the Davis Mountains Preserve, which is situated within the Chiricahuan Desert. It's part of the Trans-Pecos region, so it's mostly desert with some elevations. So near Fort Davis is the Davis Mountains Preserve, which is about 32,000 acres. And it's one of the few sky islands in Texas, which is a region that is temperate and forested in comparison with the main landscape, which is the Chiricahuan Desert. So this particular sky island, the starts about 1,500 meters in elevation, and the highest peak is about 2,500 meters. And as you can see, when you approach to the preserve, it's mostly flat landscape, but you get to the preserve and it's just this sky island essentially, where the vegetation is different, and it's a very unique place in the region. So for what we know about the biodiversity present in the preserve, plants and vertebrates have been thoroughly studied, as you would expect. But for the invertebrates, the situation is very different, where with this project that started in 2021, the purpose was to thoroughly survey arthropods and other invertebrates to be able to come up with a checklist of the species that are present in the preserve. So we have several collectors and several identifiers which come and go to the preserve whenever they have time and the preserve is open for visitors. And so would they use multiple, I guess we use multiple methods for collecting the specimens. Some of the traps are set up for a couple of weeks, and then they go and collect the samples. And some of the specimens go to either private collections or institutional collections. And so some of the methods that have been used for collecting these specimens include pitfall traps, bucket light traps, and pan traps, but we also do the typical beating sheet, netting, and things like that for collecting the specimens. So in 2023, the Museum of Texas Tech became the repository for voucher specimens. And essentially, at a meeting at the open house of the Texas A&M collection, the leader of the project, Ashley Schmitz, who gave a talk last year, he just asked me like, hey, do you want to join the project? And I'm like, well, I don't really know how to identify anything in Texas yet, but what I can offer to the project is just bring the specimens to our collection and make that data available quickly online. Because unfortunately, from a lot of private collections that are involving the project, they are not necessarily thinking about digitizing the specimens. They are just at the phase of curating the specimens, sorting them, organizing them, identifying them, and just entering all this information into a spreadsheet. So this information would not necessarily see the public anytime soon. And for some of the institutional collections, they have so many other things going on that probably this information would not make it online and available in a timely manner. So what I could bring to the project was just getting this information as quick as possible for people to be able to see it and use it. And so this drawer is one of the latest accessions that we have done of this material. And as you can see, everything is really well mounted, everything is labeled, everything is identified. So this is the kind of accessions that as a curator, I love to see coming to the collection. So all I have to do, which still takes a lot of time, is entering the information and taking photographs of the specimens to have all this information available online. So the way we do this is entering information into Ecdysis, which is the platform that we use for our data management. And we have two components in this project. One of them is a checklist where we have recorded about 1,034 species in 842 genera, corresponding to 187 species. And this is just still preliminary. People are still going in the field, identifying material, and getting new taxa recorded into the checklist. And the other component to this is that we have voucher specimens that have been deposited at Texas Tech University. And the way you can find this into the database is by using the accession number that we have for this particular batch of specimens. So if you go here, you can see that we have so far 225 specimens And if we see the checklist, we have 147 taxa recorded so far. So the way these two are interacting with the checklist and the voucher specimens interact with each other is that you can ask the database to, you can also see these records as a map. And you can see that most of the localities are in this particular region of the preserve. And you can, if you zoom in, you could see where are the gaps of information in places that are probably important to visit in subsequent trips. You can also see that every single record, we have tried to provide a set of images. At least every single record would have a photo of the specimen with its corresponding labels. And for one specimen of each identified species would have habitus pictures in high resolution that can serve as a confirmation point whenever you're not sure which species you are dealing with. So it's a point of confirmation for species identifications. So if we go back, the way this information of every single record connects to the checklist is that if, for example, we go and check and find this particular taxon, Coleorrosena fulvilaris, into our checklist, we can ask the database to show us all the vouchers that are associated with the checklist. And you can see that our species of interest is listed there. And in blue, you can see the partial information of the specimen that is associated with that species. So you can track back and forth which specimens are associated with the checklist. And you can see all the records that have been recorded for that particular species. Another way that you can see this information is by asking the database to show you, to display your taxa as images. And you can see that several of these here would have Texas Tech images or images of specimens that are deposited at Texas Tech University. So it's a different way of seeing our data. And one of the beautiful things about all of these is that this is public information that anyone can access at any point. And it has been really good for demonstrating to the people who gave us the permits, hey, we are doing something, specimens are out there, and it's not like we are hogging all this information and just, you will get to see it whenever we submit a report. So, well, some of the reasons why using ecdysis is really good for this kind of project is that it helps us keeping track of progress, not only for the taxonomic coverage of the project, but also looking at the geographic coverage that the project has been able to achieve. Since the checklist and the vouchers are publicly available in our database, it facilitates reporting and is definitely something that we can every once in a while show to the agency who gave us permits to collect there, that there's things going on with the project instead of waiting until there's some publication coming out of the project. One kind of challenge here is, again, private collections. And I think at this point, it would be good to have a conversation with them and try to convince them that databasing the collections is important. But since private collections are the effort of one individual, it's going to be, I think, tricky for them to turn into this. But I guess those are conversations that needs to happen at some point. Just the acknowledgement to the Nature Conservancy, who is the agency in charge of managing the preserve, the ones who gave us permits to collect the specimens there. And if anyone is interested in seeing material from the preserve, I recommend that you contact Ashley Schmitz, who is the leader of the project. And if anyone is interested in going out collecting, whenever we submit the permits again, we can definitely include you if you're interested in any particular taxa that might be present in there. Just reach out to us and we are probably going to be able to give you material from this particular project. Thank you. The question is if we have considered uploading to iNet. I don't know. Yeah, so I personally haven't uploaded the images of specimens yet. And I don't know if Ashley has any plans of doing this, but I think that would be a good thing to think about if it's not being considered. I'm not entirely sure if it has. Right, thank you.
And our next speaker is Losada from the National Museum of Natural History.
Hello, everyone. Thank you for coming. So today I'm gonna be talking about a historical collection from Margaret Collins, and it's a termite Isoptera collection. So Margaret Collins was one of the first African-American entomologists in this country, and also she was the third zoologist, female African-American in this country. So it's really nice for me to be able to recreate her collection that is under the Smithsonian property now. So Margaret Collins was born in West Virginia, and she passed in Cayman Island at the Caribbean Sea. Her expertise was working on termites, and mostly from all the Caribbean islands. She spent the last part of her life working as an affiliate associate in the museum. And he traveled to a lot of countries, and she also was able to take under her collection and other collections from other people. So one of the quotes from her that I like the most is this one, like, because of my family and our community, my childhood was unique. I never learned what I couldn't do as a child, as a woman, or as a Black woman. That inspired us to keep working forward on our projects or dreams, especially if you are not, in my case, because I am a Latin woman, so that feels amazing. So in this case, I'm going to talk about termites. I want to clarify that I am not a termite person, but I work in the collection management unit, supporting for the projects that we need to be worked on. So one of the most challenging things for this project was the fact that termites weren't all preserved in the same way. Like, we have a part in ethanol, like the wet collection, then the slides collection, the pinned specimen collection or dry ones, and we were having kind of fossils too and nests. And also, all the collection weren't in the same, it wasn't in the same place. So it was safe in different places. So everything started with when Floyd and I went together to pick up part of the dry collection, the slides, and some other things that appear on there from MSC, and we brought it to the National History Museum. The wet collection was actually curated few years, like 1 decade ago. So it was ready with new standards, but everything else needed to be curated. So I will say that as soon as you have the collection on your hands, you should open things carefully if you don't know what is inside, if you didn't pack it. So you don't know how is the status of the material that you are gonna be working on. In this case, then you decide how to start in order to start to work with it, you separate them and then you can decide how to start to work on it. In this case, as I say, the wet collection was already curated, so I didn't really work on it. But then I was having the dry collection to work, the microscope slides, I find out some lantern slides and the nests. I prioritized the order of how I was gonna do this project. And then, well, I will recommend to be very open-minded in how to change your technique or the recommendations you get as you are working on the project. So, assessing the condition of the collection, you can find different kind of historical preservation techniques. You can also find that they are different storage types. You can find all the material mixed up and also damaged specimens. And for that, that's another challenge, how to proceed with those. Here you can see how things were. For example, fossils. Slides were in some cases just placing disorganized into the box. In some cases, they were held by some kind of paper envelopes. But that's what I say, you need to open everything careful because you don't know how things are stored during the time. Then I was finding things like types mixed with other identified species, but that weren't types. And also nest with pin specimens, all mixed out. So I will start to talk briefly about the fossils. We find out one drawer with some fossils, and they don't belong anymore to entomology. So in this case, I just clean them fastly, kind of reorganize them, but very briefly. I created the accession in EMU, so we were able then to give them or transfer them to paleobiology that are the ones in charge of them. The lantern slides, also, I just crossed through them, organized them in just new envelopes and in one of the storage boxes, creating a label, done. Microscope slides. In this case, the challenge was mostly the different mounting media the slides were prepared. So in different cases for cleaning them, you need to use different techniques because otherwise you can destroy the mounting. And I needed to clean them in order to be able to stick the QR code or the unique identifier. Otherwise it won't stick and it will fall down. So after cleaning them all, changing techniques or realize better techniques for cleaning each mounting media type, I was able to create or place the QR code, and then they were changed from the legacy storage to the archival plastic slide boxes. In some cases, some slides were really thick. In some cases, they actually were two of them together, like two slides glued together and the specimen in the center. So in those cases, that slides that didn't fit in our archival plastic boxes we are using in the museum, I used one of the very old kind of wooden boxes, slide boxes. They were just very few of them that didn't fit on the plastic ones. About the pinned specimens, I needed to check that all the names were in the correct nomenclature before I create all the labels. And for that, I used the catalogs for being sure of that. So I used this very amazing new catalog that was written by Grimaldi and others from the American Museum in New York. It was awesome, was very easy to use. And so, yeah, creating notes and organizing everything to be ready to then place them in order, putting out what wasn't really the type, organizing the paratypes. In this case, we hold them all together in one drawer. And then also going with all the identified art and the unidentified material, placing as well one identifier QR code number in each specimen. So here you can see how they are looking with the labels, the header labels. And also drawer labels were created. Of course, while I was organizing, I was creating notes and then finally the result. In the case of the nest, yeah, I briefly kind of also organized them, place them in a better way, and that was all. So you can see how they are looking now. It was nice to find in some cases notes made on the piece of nest. And well, they were having some information. We didn't place QR codes in this case. The wet collection, as I said, was already curated, but I just want to show you how is it looking now. It's a storage not at the Natural History Museum. It's in our museum support center. For conclusion, now we know how many samples we have, how many specimens we have in total. So we are having 666 microscope slides, about 2283 identified pin specimens that are including 7 families, 47 genera, and we are holding 33 primary types and 22 paratypes. For those interested on, this is the list of what we are holding now in this collection. I will say that the recuration of a historical collection is really important because we need to be sure to preserve the specimens that they don't degrade over the time. We also are storage specimens that are getting more valuable for the future. And also with the new technology, probably we're gonna be able to take out way more information about the past, present, future, you know. I will say also that curating or making this project possible, we are keeping the Margaret's legacy continues as her contributions in Isoptera, having one of the biggest collection of termites in the world. And well, thank you very much. I'm ready for questions.
Two questions. One, in the early slide, you showed several slides that showed the barcodes on them, and a couple others that checked the cover steps. The full slide, there were no barcode or QR codes visible. Are they on the back or something?
Yeah, thank you. So question was, where are the QR code located in the slides? So as you can see, in the case that we were having a space at the front of the slide, we place it on front, so they will be captured if we do a future image. In the case that the specimens are taking the full space and we were placing the QR code, as you can see in the last one, at the back. So in that left side of that slide, you can see in the top, the front of that slice, and the bottom one is the back of that same. Yeah, no, it's fine. I also forgot to say it, so thank you very much.
Standard boxes. Do you have any physical boxes where the slides went? That's where it would have gone. Did you have any kind of physical notation in the slides elsewhere?
Well, question was if we were having an extra protection for the slides that were thicker. Well, when I was trying to place slides on the plastic boxes that we use for storage, some of them didn't fit because of they are thicker, sorry. So I was leaving them in a side, trying to find out if we were able to find a plastic box that were able to hold them. As we didn't find, and we tried many cases, we decided to go back for the really, really old ones. They are blue color outside, wood, books the same, but the spaces are way bigger, so they fit perfectly in there, so we didn't really need to put any extra protection.
Slides very thick. What you're saying, as opposed to a large distance between the back and slide in front of the person.
Well, they were totally in good state. Like they were just, the person that prepared that slides made them twice of them for some reason that I don't know. So instead of use a cover of the slide, they used 2 full slides. So they were double. Yeah. Well, you know what? They weren't really specific, they weren't organized by nomenclature because we weren't covered that part, and I am not a specialist. I am not a termite person, I work on Lepidoptera. So in this case, we just save them to preserve them correctly. We are sure they are following all the standards. And then if we get a specialist for it, that person will have all the material ready to go and work on the real nomenclature classification. We don't have actually a curator that is an specialist on Isoptera in the museum, so. Sure, you're welcome. Thank you very much.
Our next speaker is Megan Neal from the McGuire Center.
All right, hi, I'm Megan Neal. I'm a research assistant at the McGuire Center of Lepidoptera and Biodiversity at the Florida Museum of Natural History. And my job consists of microscopically capturing and processing high-resolution images of Lepidoptera genitalia. I have to say it's kind of nice talking to a crowd that actually thinks of this as a normal thing to do. It's Thanksgiving of my in-laws this year, and I'm expecting lots of raised eyebrows and some, oh, that's nice responses. So many of you probably know that Lepidoptera genitalia are uniquely shaped in each species, and that high quality images of these genitalia can be a very useful and low-cost taxonomic tool. So today I will briefly go over the significance of genitalia dissection and analysis in Lepidoptera taxonomy, using an example from the genus Dryadala. after which I will provide an overview of the McGuire Center slide collection. And finally, I will take you through the process of digitizing these slides. Now, many of you are probably aware that damaged and degraded specimens are inevitable when using collecting methods such as malaise traps or sticky cards for long-term monitoring in inaccessible areas, or for just trapping moths that don't readily fly to light. Figure 1 to the left depicts a petri dish of specimens we have dubbed moth broth from a suction trap into alcohol. This gives you an idea of the condition of the specimens that are collected in these types of traps. It probably also gives you somewhat of an idea of how wisely I use my time on Photoshop. It's in cases like this where genitalia analysis really becomes indispensable. Even when the specimen is rotten and the DNA is degraded, right? The genitalia are often the only parts that are recognizable. And comparing the genitalia of degraded specimens to dissections from authoritatively identified specimens allows lepidopterists to make determinations in cases like this. Besides facilitating identifications for degraded specimens, genitalia dissections can also be really useful in helping lepidopterists differentiate between externally similar species. Figures 2, 3, and 4 to the right there depict 3 native species of Dryadala, and figures 5, 6, and 7 depict their corresponding genitalia to the right. This genus seems like an extreme example because the genitalia are really asymmetric, so it can be hard to tell what parts are what. So the dissections kind of look like someone had a rough day in the lab, but they are actually interspecifically consistent. The first species is Dryadala vesaliella octurum. True D vesaliella is a temperate species up north. And this here is species number 39 in an unpublished manuscript by Don Davis. The second species was dissected from traps in northern and central Florida, and it's tentatively named for the collector who got them in beetle traps. And as you can see, both are dark gray in color. They look very similar. Last year, museum volunteer Bob Belmont caught Dryadala specimens in a Ceciad pheromone trap, and species 1 or 2 were expected, but of course, when they were dissected, they were found to be a third species. All right, so genitalia dissection is an important taxonomic tool. We got that. So let's talk about the McGuire Center's slide collection. All right, so the McGuire Center slide collection, it's an extensive collection of over 20,000 genitalia slides, with more than 8,000 of these representing species that occur in Florida. In the photo to the right, former museum technician Kevin Burnett can be seen sorting what's estimated to be about 1% of the slides housed by the McGuire Center. And the center also retains numerous slides containing various other structures like wing venation and larval cuticle and head capsules, as you can see in figures 8 and 9 there. You may have noticed that these are from specimens of undetermined species. It's because priority is given to slides like these that contain structures from specimens needed to resolve the taxonomic status of newly detected species in Florida. So I've thrown out a few numbers, but the McGuire Center slide collection is actually continuing expanding. In addition to slides donated by external sources, new slides are prepared each year by McGuire staff as new specimens are collected. So this is a growing and fragile collection, and that's prompted the center to digitize slides primarily for use in research publications, but also as a public resource. So the goal is to capture detailed composite images of slide contents like these here for most specimens that occur in Florida, and then upload them to the museum's online database Specify. So this digitization project is funded with a grant obtained through the Florida Biodiversity Foundation, and it has 3 advantages when compared to the other available platforms on Lepidoptera genitalia. As you can see, the images are very high quality. This female genitalia of Tritricid species, Sparganothus sulfuriana, holds a special place as record number 1 in our Specify database. Furthermore, Specify is an official specimen database, so the user can access all the data on the specimen. And finally, Specify is the official curatorial database for the whole of the Florida Natural History Museum. So as an institutional website, it should be supported for the long term. All right, now we have some background on the McGuire Center slide collection, so I'll take you through the digitization process. So slides with a little less potential for publication are scanned on an Epson Perfection V500 flatbed scanner at a resolution of 2400 DPI or dots per inch. The image in the red highlighted bubble shows an enlargement of the slide when scanned at 2400 DPI. And some of these are then that have more publication potential are just later selected to be fully digitized as shown in the above full resolution genitalia image, sorry. All right, so I'm going to demonstrate the process by which we produce images like this one up here. All right, so first images of the slide mounted moth genitalia are captured at 10x using a Leica DM6B microscope equipped with a camera. So our goal is to capture detailed information across the entire specimen. And to achieve this, we acquire a stack of images for each slide. And stacks are just created by capturing 20 images at different Z planes covering the entire focal depth. And each image in the stack represents a specific focal plane of the specimen. I think the diagram to the left depicts this process really well, actually. The microscope captures multiple images across the XY plane and then stitches them together to create a seamless image. And it then repeats this process at various Z focus planes to form a complete stack. All right, so we have our 20 slices representing the full range of Z planes, and then we use Zerini Stacker to produce a final composite image. And Zerini is just a software used for focus stacking, and demonstrated by the diagram of beetle figures to the right. Focus stacking is an image processing technique that just aligns and merges the in-focus areas from each image in the stack to create a single image with extended depth of field. So the next slide, I'm just going to play a short video of Zerini processing the 20 images we just acquired. On the far left panel, you can see where I have dragged and dropped the files consisting of my 20 slices. And then the image you see up there now is just one of 20 slices, and it corresponds with the blue highlighted file on the far left. And then when I play the video clip, you'll see me select the DMAP stacking algorithm to begin the stacking process. And then on the far right, a panel will appear displaying the current output image, which will become sharper as the focused areas of the slices on the left are incorporated into one composite image on the right. All right, so I begin the stacking process. Zerini starts with a slice and sequentially moves through the next 19 slices, taking the in-focus areas of each slice and incorporating them into one composite image. And just notice how at the end, how areas such as the tegumen and phallus are sharper in the composite image on the right when compared to the last slice on the left. All right, so this is the final output image produced by Zerini. And as you can see, all areas of the genitalia, including the CD, are in focus. But of course, there's some further processing that needs to be done to remove the more obvious dust and dirt artifacts present in the image. So I'll go over some of the basic techniques that we use to clean up our stacked image using Adobe Photoshop. All right, so many of you probably know that programs like Adobe Photoshop and GIMP are software for image editing and manipulation. But like me, before I started working on this project, you might not have that much experience with it. So to familiarize you with programs like this, using Photoshop, I'll present a few short video clips of various widely applicable basic techniques that can be used to clean up genitalia images. So just keep in mind, though, that each user develops their own preferred set of techniques and tools to achieve their goal. So there's not really one way to edit an image. All right, so in here, I start with a white background layer and create another layer with the stacked genitalia image. And layers just allow you to separate components in an image and then work on them separately. So in this case, I'm simply creating a white background layer overlaid by the genitalia layer. And once I have these two layers, I use the object selection tool to obtain a rough selection of the genitalia itself. And I say rough because Photoshop has limited capabilities in detecting objects with complex edges like we get with the genitalia. And as you can see, it missed large areas of tissue and CD and selected areas within the genitalia that we don't want selected. Just importantly, the object selection tool gives us a solid foundation from which we can adjust our selection to better fit our needs. And then we can make these adjustments using the polygonal lasso tool. And this is just a type of freeform selection that utilizes straight edges. And when the shift and alternate keys are selected in conjunction with this tool, you will see a small plus or minus sign appear next to my cursor, just indicating that we can respectively add or remove areas of selection. Both functions are demonstrated here, in which I'm first modifying the selection to include the whole CETA, and then modifying to exclude the unwanted artifact interfering with the CETA. All right, and then here, skipping ahead, I've already made all the alterations needed to complete my selection of the genitalia using the polygonal lasso tool. And at this point, I used the select inverse function to select the background instead of the genitalia itself, so that I can remove the original background and expose the underlying white background. So we have a clean white background. However, more processing might be necessary, internally or at the edges of the genitalia. And the selection and background process sometimes results in a hard edge where there should be a soft edge. And we don't want the genitalia to look like a cutout. So I use the eraser tool to just delicately remove parts of the genitalia layer to leave a soft edge where the polygonal selection may have left a hard edge, or maybe some remnants of the original background. Okay, and in this video, I used the clone stamp tool, which enables you to copy or clone an area or apply it elsewhere. This just allows me to sample the areas adjacent to an unwanted object, whether it's debris or some other element, and apply it elsewhere. This has to be done pretty carefully and generally on a small scale at high magnification, because you don't want to duplicate an area that conspicuously doesn't belong or adulterate the image with artificial and misleading features. And you can see me change the size of the cursor a couple times to fit my needs. At the end, I also used the blur tool lightly, and this is like the eraser tool, just without the deleting effect. It just homogenizes and naturalizes the cloned area and softens the edges of the outer piece of tissue there. And in this screenshot, you can see the fully stacked and edited image. The area surrounding the genitalia has been removed for full exposure of the white background, and the genitalia itself has been edited to remove any unwanted artifacts. And then just for comparison, the stacked image before editing and the stacked image after editing, with some of the blemishes I removed highlighted in red there. All right, and finally, we have our finished composite image, including the 600 DPI slide scan overview, the scale and image identifiers. And this final stacked and processed image is then entered into Specify. As I previously mentioned, Specify is the museum's database for biological collection. And so far, about 700 full resolution composite images, as well as 2,200 slides scanned at 2,400 DPI have been completed and entered into Specify. It's pretty modest, but with priority given to undetermined species and Florida specimens, the McGuire Center has been able to advance some of its current research. Another approximately 38,000 entries have been made by McGuire staff containing collecting data and images of pinned adult specimens. So as we move forward, we want to continue expanding on the specified database and just focus on producing high quality images to support ongoing research and publications. And we hope that by digitizing the collection to ensure that our collections remain an accessible and vital resource for the scientific community. All right, thank you very much.
All right, now we'll have a brief break. See you back here at 10:10. And just a plug for the silent auction, please, proceeds to the ECN Foundation. Stop by, put a bid in, best price on some really valuable books, and again, proceeds to a good cause. We'll close the silent auction just before the lightning talks. So get those bids in.
I don't know what it is. Good luck.
Come on,
Come on back.
most important talk of the whole conference.
You heard Mike. Our next speaker is Mike Ferro from Clemson University Arthropod Collection.
All right, am I up? All right, well, thank you very much. Hello, everybody, the ones of you that are here. I'm Mike Ferro and Natural History Literature Rescue. It's a 501c3. My wife is the one that did the paperwork. She has superpowers. There are 3 people on the board. Myself, Victoria Bayless is treasurer, and Anthony Dzinski is secretary. We have 7 places available on the board total, so be careful. We've had a lot of conversations about what a collection is and our work. And so I just want to point something out. People who are born rich are not ashamed of it. You have an awesome job. Don't be ashamed of it. Collections are tools, beautiful, beautiful tools, and you get to build them and work with them. Don't be ashamed of that. And collections of literature are beautiful, beautiful tools. Build them and love them. If you're going to be a scientist, if you're going to be a researcher, you will build a library, whether it's physical or in PDFs, and you're going to have to work to maintain that. You're going to have to buy a new computer every 10 years, and you're going to have to get subscriptions to different things, or you're going to have to lug heavy books and papers around when you move. So appreciate the tools that you work with. Don't be ashamed of them, and appreciate the situation that we're in. It's absolutely amazing. Also, the artist, Caitlin Silva, is looking for a PhD. She's fantastic. And she will be at, some of her art will be at ESA. So look for her. Okay. Natural History Literature Rescue. We work to preserve literature, specimens, samples, equipment, supplies, memorabilia, and ephemera associated with the study of natural history that would otherwise be destroyed or lost if not maintained. This is a very wide net. And it's not entomology literature rescue, it's natural history. The reason that this is written up this way is because a few years back, we were asked to come in. A researcher had passed away. He had a very extensive home laboratory. His wife didn't know what to do with it. And so we hauled off an enormous amount of stuff. And so this was originally formed largely to save reprint collections, but I realize that we're going to have to cast a wide net. Also, for those of you that are out there, most of you will appreciate this, but I'm going to have to tell people, if I start a foundation to save puppies and kittens, if you hand me a kitten, you've given me a burden. This is not a gift. Yes, I know we were trying to save them. And don't hand me a puppy and say it's worth $2,000. If it's worth $2,000, sell it. We're trying to save things that are going to be thrown away. So please appreciate that. You may have a lot of science or National Geographic or something like that. That's lovely. I want cash or your home. But yes, anyway. So right now, what do we have? These are largely a contribution from Tommy McElrath gave us Ed Mockford's Socotra reprints, and I've hauled a lot of stuff to giant trailers from Florida Collection of Arthropods. We've got, so every foot is about 175 papers. Well, every foot is about 175 papers. We have about 25 file cabinets full of material right now. You'll see that we've got 2 feet of, camel spider literature. Spend some time trying to collect and put that together. We've got about 1,750 coccinellid papers. That's a pretty good chunk. We've got almost 3,000 pseudoscorpion papers. I have not got to look at most of these, unfortunately, but just thumbing through, we have original dissertations from Slovenia from 1972. I mean, these are magnificent collections because the people who were building them were not just getting material themselves, they were having material sent to them. We have personal communication there's a lot of history there. And again, it's a magnificent tool that can be used. And people put this together over, in some cases, generations. We have, what was it? So Weems, Myriapod, and Crustacea, that's actually multiple. I don't know if it's all of Weems because there are multiple other collections that he obtained or that were obtained after him. And so these have a really beautiful, beautiful history. Approximately 32,000 documents is what we think we have right now. And this is just the tip of the iceberg. I'm actually incredibly frightened about what the future will bring. But anyway, for the fun of it, in a file cabinet that was going to be thrown away, this is the sign-in sheet for the 1936 Entomological Society of America meeting. This is the original. If you want to touch it, clean your hands first. This was literally in a building with no pest control, no air conditioning, no in Florida. When you run through this, 1936, Snodgrass was there. And Edith Patch, she was the first female president of the Entomological Society of America. She was an aphid researcher up in Maine. There's a magnificent biography of her. Frederick Poos, inventor of the pooter. Some of you incorrectly call it aspirator. And I was running through, this is a fun one, Marjorie and Loris Milne. So they met at Woods Hole in 1934. They were married in September of 1936. This meeting took place in December. They were, if you'll notice, she was in Maine and he was in Texas. They didn't get to live together for another 5 years after they got married. Once they got together in New Hampshire, they became a power couple. 55 books. I immediately got on Advanced Book Exchange and started buying as many as I could. They're fantastic. And over 100 articles. So there's a lot of beautiful history here that would have gone in the trash. And so I'm not doing a hard sell here, but I'm trying to point stuff out here. So I know that we're just, let's save some papers that nobody cares about. I'm a trash collector, but I can dream bigger than any of you. And eventually someday it would be nice to have a building and maybe a monorail. And if we get enough, some cloned dinosaurs. You laugh. For right now, we have no money. So the idea here is to stay small. I've covered all the expenses so far. We don't have any monthly expenses right now. Although again, I'm scared that tomorrow we're going to have a lot. But my idea is to keep as small as possible to push these things away and make sure they're not destroyed, but not spend much money on them, storage containers, etc. I'm currently using some, Clemson is a silent partner. So silent, they don't even know. Advertise. I hear horror stories of these things being lost. And so if we can let people know that there is interest, advertise to everyone you can. I'm going to hit all of the other groups if I can, the geologists, the ornithologists, maybe they've got a lot of money, I don't know. The mammalogists, the mycologists, hopefully we'll find somebody with a couple of million that would, turn this into something. And then also get money, lots and lots of money. Now, I am incredibly risk averse. So if you give me 100 grand tomorrow, it's going to go in the bank and we're just going to get interest on it for a while. I am not interested in buying a place and getting a secretary and I want to make sure that we don't make mistakes. This is a long-term progress situation. I also see this as a community resource. Clearly, I don't know what I'm doing. Go to the webpage, look around. Those of you that do know what you're doing will notice that we're missing some things, and I'm aware that we're missing some things. And you'll also notice that I am not doing a hard sell. I'm not going to give you a QR code at the end of this. I'm going to give you a webpage. You have to type it in. And on the front of the webpage, it doesn't say donate. You have to go up and click the little tab to donate. So I'm not going to be any good at this, but I'm not going to hound you because I've been hounded by a lot of places before. So we'll see what happens. But I would love for people with expertise, with knowledge. Again, we have seats on the board, or if you just want to volunteer or help, with something, that would be wonderful. It is a community resource. I'm not interested in this being some sort of a fiefdom. So it is a long-term project. Eventually, it'd be nice to have a building, a place where people can show up, a place where we can have volunteers. Digitization at Clemson, we have about 11,000 reprints already. I took a photograph of the top of every one of them, and it's all online. So you can flip through our collection. A lot of those papers have been digitized. They're on Biodiversity Heritage Library. So there's no need for me to digitize them. There's no need for me to worry about all the legal issues of digitizing and holding them when somebody else has. But if I have a huge reprint collection and I have, hopefully through fair use, shown you what we have, then somebody can find that. And if we have volunteers or even an employee someday, they can email in and say, hey, I need this paper, or I want a higher resolution scan. We have posters in the back, outside, and I have the original of it as well from Anthony Dushinsky, of the first the first American entomologist. Go to Biodiversity Heritage Library, look at their scan, look at our poster. You'll notice we have details they don't. And so I'm imagining a future where people will be able to get high resolution scans of those images that they need. Yeah, all right. So at this point, I don't know how much time I have. I'm happy to answer 2 minutes. I'm happy to answer any questions. And like I said, I have originals and I have, this is a PDF on the web page as well, if you go looking for it, if you want to download it. And I haven't even gone through all the names yet. So, yes.
Yes, I wonder how you're gonna, if somebody wanted to donate literature, a lot of it, how are you gonna pay to get that shipped out where they are?
Okay, so the question, yeah.
So how are you gonna prevent constantly getting duplicates of everything?
Okay, so the question is, how are we going to get the literature if they want to donate it, and how are we going to get duplicates? First of all, I love to drive, but part of the reason that we set up the 501c3, and trust me, it's difficult to do this, but part of the reason is because I wanted to have a situation where I could email out on the listserv or other places and say, this is the literature, this is where it is, this is how much it will cost to get it to us. Is anybody willing to donate? And then they're not donating to Mike, they're donating to a real organization. With a 501c3, we can get grants too, if people want to write them. And then duplicates, I'm not worried about duplicates because in most cases, what I'm getting is an entire collection. I'm getting everything that someone put together with their notes, their annotation, those individual papers are associated with other papers. And if I get a duplicate over here, I'm not so much worried about it. It would take more time to go through, and I'm not interested in throwing those away. So we are going to have duplicates across the spectrum. Oh, Mike Ivy in the back, just yell. It's not a bad idea. Have I considered getting UN money for carbon sequestration? So anyway, yeah, like I said, for those of you that are interested in helping out or donating materials, or better yet, money, please consider. And like I said, I have an original if you want to look at it, and I have some copies if you want to look at those. Thank you very much.
Our next speaker is Krystal Hans from Purdue University.
Well, thank you so much. And I do have to thank Andrew for the invitation to speak to this group. This is my first ECN meeting, and this is such a welcoming group. So I'm really excited to share some information about my field of forensic entomology, so a very niche area within entomology, and some of the concerns that I have about the future of our information and our evidence. So just a little bit of background, I will not be showing any graphic photos. I won't go into case details or anything like that for this group. Ask me later, I'm always happy to share those and give you some horrifying nightmares to take home with you. But in my field of forensic entomology, we're looking for ways to apply information about our insects to different applications, whether it's civil cases with urban or stored product, veterinary cases of animal abuse and neglect, or wildlife poaching and trafficking, or in my world, about 99% of the casework that I get is in medical legal, so dealing primarily with death investigations. Now, the way that I'm using insects, and I know I don't have to sell this to this crowd, which I have to say is a refreshing change. I gave a presentation yesterday to law enforcement, and I always feel like I'm a bit of a salesperson giving a pitch about why you should care about insects. So in death investigations, the information that we can provide to law enforcement is about our insect behavior and activity. And so we call this the time of colonization. So when did our flies arrive? I jokingly refer to my flies as the first responders. Police officers and firemen usually cringe at that, and they're a little offended. And so when those insects show up, they are able to deposit their eggs, and their offspring are the ones that are feeding on those remains. And so providing information about that time of colonization, when we know that our insects arrive pretty quickly after death, and those offspring that are feeding on those remains, being able to identify and determine what life stage and how old those insects are, can provide a lot of information about an estimate of a minimum time since death of that individual. So different ways that we can use our insects to get some pretty significant information. Now, I know that I don't have to describe to this audience about the kind of amazing world of insects, about their ability to detect those really noticeable smells of decomposition, about the impressive power of maggot masses and maggot feeding, and how much food they can consume. But I generally have to give all of that information to law enforcement. Now, compared to your area, I work with a pretty large group of all different people that generally don't have a background in entomology. And so in my kind of case management, this is the best way for me to be able to summarize what we do at all of the different steps throughout this case experience. And so I often get a phone call, a text message, an email from a variety of different people asking me to consult on a case. And so this could be a crime scene investigator. We have a coroner system in our state, which is a challenge in itself. Could be a death investigator, a detective, or sometimes insects are not observed on the remains until they go to autopsy, so a pathologist or a medical examiner. Now, when I get a request, I always like to be available for the scene as long as I can make it, and I don't have to cancel class to do that. If I can attend the scene or the autopsy or both, that's preferred for us because as entomologists, we know what we're looking for, and we like to make sure that we're not missing any really crucial evidence. And so at the scene and autopsy, I work with another set of people, again, that do not have an entomology background. Now for the analysis component, so once we've collected the evidence or the evidence has been shipped to our lab, we're providing information about that analysis, about the identification and the timelines that we generate to the detective, to the pathologist or medical examiner, or to attorneys sometimes directly. Now, the issue that we have sometimes in our field is that all of the hard work that we do to provide this information for a case sometimes sits, sometimes for months, sometimes years, sometimes for decades. And so by the time a case actually goes to trial, we are now sharing our science with the public, with a jury pool of people that usually have no concept of what the word entomology means. And so it's usually a pretty big undertaking for us to be an expert witness, to be able to provide this information in a meaningful way to explain all of the work that we did for that case. Now, I'm a board certified forensic entomologist through the American Board of Forensic Entomology. That is the only certification board that we have for our field of forensic entomology. And in our field, we've done a lot of work to generate standards for collecting our insects, for how we can make sure that evidence is properly shipped so that it's secure when it arrives to the lab, how to rear our insects. And our field is growing a little slowly. We have 19 board certified forensic entomologists and 4 that have emeritus status. That's a pretty small group of people. Now, if we look at this across North America, this is a heat map of distribution of where we're located. So I'm in Indiana. You can see Texas always does things much bigger, right? So we have quite a few, I think 5 board certified forensic entomologists in Texas. My colleague Lauren Weiner at Arizona State. We have a couple in Ontario, one in Florida, West Virginia, New Jersey, and one out in British Columbia. So we're kind of widely separated geographically and try to offer our services to as many different agencies as we can. Because there's so few of us, we sometimes get stretched kind of thin with our work. Now, as I said, we have a lot of standards for how we can collect, and this is kind of the general workflow that was provided by the American Board of Forensic Entomology for things to consider when you're collecting data at a scene or at autopsy. So making sure that photographs are taken. Can't tell you how many times I asked law enforcement, oh, did you take some cool photos of the insects? No, we didn't even consider it, right? So it often gets overlooked. Temperature data. So having all of that temperature information about our insects at the scene and in the location where the remains were located. How to collect our representative specimens, how to make soil samples and debris collections to make sure that we're not missing anything that's crucial, how to preserve and rear, what to do at autopsy, and how to ship samples. So we have all of that information and we have that standardized. But what I see is that we have this incredible number of unresolved cases in the US. And just a couple of statistics, we have 340,000 unresolved homicide cases just in the US. And what we see with law enforcement is that we're now dropping below a 50% clearance rate on those cases. My question is, what happens to the entomological evidence for these cases that are unresolved? Evidence that might have been collected, and maybe we're in that waiting period between when a forensic entomologist might have collected, might have examined, maybe they wrote a case report, and nothing's happened with this case and it hasn't moved forward to trial. And so I started thinking, well, what are we doing as a field to try to deal with this issue? And so I started asking some of my colleagues who are board certified. And my first question was, how are you storing your forensic specimens? So I know a lot of the presentations here have shown incredible things that you're doing to be able to digitize and take incredible images. The last presentation was about retaining information that could otherwise be disposed of or lost in time. And so I think that that really segues nicely into what I see in my field. So I asked, how are you storing your forensic specimens? And so I found that about 90% said ethanol. Okay, so they're storing in ethanol. We had a range of different percentages of ethanol. I heard anything from 75% all the way up to 95% ethanol. Some people are storing all of their specimens in ethanol, whether it's maggots or it's adults, flies and beetles. Some of my colleagues are freezing their specimens. So they're keeping reared adult insects in the freezer. Now, I'm sure most of you in this room have had an issue with a freezer at some point, right? Where it's failed and we could have damaged specimens. Only about 45% of our colleagues are actually pinning any specimens. And about 10% said that they're keeping them dry, meaning that we have piles of beetles and adult flies dry in a vial sitting somewhere in someone's lab. So this was a little alarming, right? We don't have a standard way that we're documenting and keeping our specimens. Now, how about retention of specimens? My expectation, what I do, I keep them forever in the event that somebody might need them in the future. I found that a good portion of our colleagues were keeping them indefinitely. Some of them had different numbers of years that they were retaining them, whether it's 10 years, 2 years, 1 year, or they were immediately returning them to law enforcement. Now, my next question was about, now what happens when you retire? So this is for our emeritus colleagues. 50% said that they returned all of their evidence to law enforcement agencies, whoever was in charge of the investigation. Now, I've already commented that law enforcement, they're not entomologists, right? So they don't know what they're doing with that evidence. Now, this is probably going to break my heart more than anything. 50% said that they had disposed of all of their evidence upon retirement. Now, to me, that brings up a lot of concern. So I have been in several evidence and property rooms for all different agencies across the country, and I've seen the state of how they're maintaining their evidence. They do not have proper storage for our insect evidence. They are not routinely checking on evidence until it's ready to go to trial. If there was an opposing expert that needed to review this insect evidence, that can be a challenge. And sometimes there's a lack of documentation and chain of custody. And so my concern for the future of forensic entomology is that with the number of criminal cases that we see a large percentage that results in a plea bargain, meaning that they will not go to trial. But in federal court, there are about 50,000 cases every year that are appealed. 7% are overturned. At the state court level, it's a much higher percentage, about 15 to 20% of these cases are overturned, meaning that somebody is responsible for reviewing this evidence to be able to advocate for that case. Now, I've had several cases where the agencies have sometimes requested the evidence back, and I ask, well, what is your protocol for maintaining insect evidence? And the response is usually, well, it just gets stuck on a shelf with the rest of the evidence. And nobody's paying attention to that. So the reason I'm here today is I kind of want to make a pitch. I think that this is the greatest group to be able to do this. I think that the future of forensic entomology, to be able to retain our evidence to be used for future appeals, for potential wrongful convictions, or some of these unresolved cases that haven't yet had any leads, we have to have a policy in place to be able to retain them and make sure that they stay with entomologists, to make sure that we have photos of all of this information, that these specimens are properly curated and databased. And if we have a partnership with museums that are willing to let us house some of this evidence in the long term, particularly for forensic entomologists that are retiring, so that this evidence is not being lost and that we can still use this in the future for cases. I know that I've gone over my time. If you scan that QR code, that'll bring you to my electronic business card. Happy to work with anybody. I have not yet talked to Chris about the perk taking my collection when I retire, but that's in the works. Happy to take any questions if anyone has any. Yes, I will.
Do you think that law enforcement or any of the other agencies would kick some funding to the collection to help maintain their evidence?
The question was, do I think that law enforcement or another agency would be able to fund safekeeping of that evidence? Yes, I do. So they pay plenty of money to be able to store their evidence and to be maintained by an evidence technician. So I think that that's a fair ask to have them fund that maintenance. Mm-hmm. Oh, absolutely. Yep, I'll be here. Yep. Okay, thank you.
Our next speakers are Jessica Bird and Erin Kolski.
Hi, thank you all for coming. Before I get started, I just wanted to say that this talk was created by Erin Kolski. She was supposed to be here to give it in person, but was unable to join us. So a big shout out and thanks to her. So the USNM Odonata collection, obviously, as you all know, it contains dragonflies and damselflies. We have over 115,000 specimens, and it represents 3,265 species. It's one of the largest and the richest Odonata collections in the world. This collection was curated for a long time by Ollie Flint. After his passing, Torsten Deco has since become the curator. And so you'll hear both of them referred to a lot in this talk. As you all know, pen specimens can take up a lot of space, especially when we're talking about like well-curated spread dragonfly specimens. So to help take care of that, most of our specimens are actually stored in flat glycine envelopes. This was a curatorial choice that was developed around the 2000s. Many of the penned specimens were relaxed, flattened, and then these index cards that you can see on the right were created by Ollie Flint, where he actually typed up locality details and then placed them behind the specimen. That kind of gives the flexible envelope a little bit more support. When there were traditionally penned specimens, those original labels were kept on the back, so they're kind of slid behind there in case any information didn't make it onto the type card. Since then, anything newly collected is curated in this format, and no traditional labels are used. We just type it up directly on the archival cardstock. As you can see, there's about 20 dragonflies that can fit in a pin drawer. In one of our boxes that we now use, you can fit about 100 dragonflies. We still have 4,000 to 6,000 that are pinned. These are type specimens, and also rare or specimens that would have been completely destroyed had they been removed from their pen. So one of the cool things about the Odonata collection is that it was one of our original specimen level digitization projects, or one of our first specimen level digitization projects. This was kind of a labor of love. It took a lot of people to get it to get this project even started. When they did the original databasing, we used USNM barcodes. As you can see in the picture, these are kind of like a flat barcode. Now we still use the same numbering scheme, but we use a 2D barcode. At that point, all of the data was placed into EMU, which is our collections museum information system. And also georeferencing funds were sought. In the early 2000s, this was a lot of money and effort, and only 10% of the collection was able to be georeferenced. It is our only complete order in entomology with a full specimen level database that is available publicly online. So because all of these insects were already individually databased, they lended themselves well to a quick imaging project. So in 2022, our herbarium was wrapping up their own full collection digitization system. They were utilizing a conveyor belt, which many other herbariums have used. It's a system that was developed by Picture. Torsten Deco gave a talk on the imaging of the Odonata specimens a few years ago. So if you have any questions about how we actually imaged and what those individual photos look like, I would be more than happy to send you a PDF of his talk. But basically, since the enveloped Odonata specimens are flat, much like herbarium sheets, we were able to modify their workflow and use the exact same imaging system to get these very high quality photographs of each individual specimen. It was kind of a really cool project where we were able to use something that was developed for botany within our museum and take that and, make it work for an entomology specimen. And at a time when our museum was talking about silos and how we all needed to work together, it was really nice to see this project that was able to span other departments. So these images were all labeled at the time of taking them. They were all titled with the USNM Mint number. And this became particularly important when we were incorporating them back into EMU. So due to the file naming schema, we were able to add those very seamlessly, attach them to our already databased specimen records to give us this fully databased record. All of our, almost all of our images at the Smithsonian are labeled CC0. That means that they're in the public domain and anyone can use them. There are a few cases where we don't do that, either if the specimen is endangered or if there were certain, if the photographs were taken under a certain sort of contract. But for the most part, we try to make those, all of those images 100% in the public domain. Since we had fully imaged specimens, we were able to enrich the digital records. So now we have, a picture of the data. We're able to double check some specimens that may have had fuzzy data or showed up on other sites as possibly like a locality mismatch. And we were able to really enrich and delve into that data that was originally collected in the early 2000s. We also added GWIDs for some of the people involved. This came up in a previous talk, but adding GWIDs to collectors and authors can be extremely time consuming. This is not the main focus of any of our projects, but if there is a collector that tends to come up a lot, we'll try to add a GWID so that they are getting direct credit. We also continued georeferencing. Now over 96,000 of our specimens have been georeferenced. That's 80% of the total records. We're also including some that have data points that cannot be georeferenced. To the right, you can see a map of the Odonata specimens in GBIF, how they've been collected all over the world. We still need to work on some specific areas. We're lacking georeference points for China, Indonesia, India could use some work, but we are slowly trying to finish the entire collection. Also, this has reduced loans. So since we have imaged all of our Odonata specimens, we have not had any loan requests for this collection. Now, Odonata was not a highly transacted order, so it didn't, I wouldn't say that that would be the norm for any other orders that we fully digitized, but it has greatly reduced the requests. Another thing that has happened is that students are able to look at our specimens, and we are in the talks with several students that would like to select specimens for DNA analysis so they can see which species we have, exactly how many, and which specimens they would like to use. An additional thing that has come out of this project was the adding of location data. So this is separate from obviously the locality data that's found on our collecting cards. I'm talking about the location data of where the insects live in the collection. So in 2024, we started moving the specimens into these new boxes. Whoops. Yeah, into the new boxes. And instead of the old kind of cardboard boxes that we had with stickers on the front, these have removable pockets that allow us to change the headers. And so with these new boxes, we are also adding a barcode. It's not in that picture, but they will have barcodes. And so they can be scanned and you'll be able to find the exact place of where they are in the collection. There are a couple of other collections within our museum that also do this, but this is the first one that's been put into EMU so that visitors would be able to log in online and actually find them without having to consult somebody. And this really helps to improve the accessibility of our collection. So what's next? We are going to complete the rehousing and the location mapping. Sorry, this is the picture that I was talking about. We have these like older boxes right here on the right with the stickers, and then we have these newer archival boxes. We are going to continue imaging the newly returned Odonata specimens as well as the penned. Obviously, since this project started 25 years ago, there are a lot of people to thank, and not everybody involved was listed on here, but it wouldn't have been possible without the help and the funding of a lot of different people. So thank you so much. Okay.
Amateurs, are they curated by an entire different unit, alcohol unit? Are there plans to do like a digitization?
Thank you. The question was, do we have plans to image or to cure, or do we have plans to database the immatures that are stored in alcohol? So the DIPTR unit has an alcohol room, and we actually were just talking about this. So there are hopes to digitize the alcohol collection, but it hasn't happened yet. So we're going to start with the types, well, with the types of the DIPTR that are in alcohol, and then hopefully we'll be able to do like some of the immatures. I do not know. I could get back to you, but. Oh, that is a question for Floyd Shockley, who is not here. Are the new ones from HH Elements? Okay. Dave would also know. Sorry, I typically do the data. I don't do any curation or ordering of the supplies. Thank you.
And up next is Samanta Orellana from ASU.
Okay. Good morning, everyone. Thank you for joining. My name is Samanta Orellana. I am currently affiliated with Arizona State University, but I defended last Friday, so that will change soon. Thank you. So I work with this fantastic group of beetles called the fungus weevils, the family Antribidi. I have been working luckily at ASU using the O'Brien collection for the last 5 years. So I have been able to check specimens from all over the world. The O'Brien collection has over 8,000 specimens from everywhere. They collected around the globe. So I was very lucky. I had specimens to check. So I focus on the neotropical region, where around 15% of the species are currently known. But we have over 200 species that are currently undescribed. So there's still a lot of work to do in the neotropical region with anthribids. During my dissertation, I worked only with collections. I had over 3,000 specimens just in the O'Brien collection, plus the loans I got from other institutions. So it was really interesting to have this global approach on the group and being able to check a lot of specimens from all of these collections. So I ended up with 100 species of anthribids that I was checking from the neotropics, the two subfamilies that were recorded, 15 tribes, 49 of the 61 genera recorded, plus 4 new, we have more, maybe 6, 8, a lot of new groups over there. So I also have I was lucky enough to get all the type genera for all the tribes recorded in the Neotropics, and also the type of the tribes that have genera recorded in the Neotropics, but they were originally described from elsewhere in the world. So I was able to get all 3 subfamilies, 16 of the 33 tribes for now, 63 genera, and 128 species of Amphibidae for my analysis. All of these specimens are right now in the O'Brien collection. So it's a really, really nice collection to work with, plus a couple more specimens that I got from somewhere else. So I got a phylogeny. You will be reading from this about this very soon, but with lots of morphological characters, very nice. But today I want to focus on all the work that was behind picking up all the taxon sampling that I used during my study. So during this time, I published a few catalogs for the Neotropical region, Panama, Guatemala, Colombia, and while I was checking the specimens and choosing all the groups that I was working with. I had to identify a lot of specimens, and I was working also with the digitization of this material so it could be available for everyone else. So today I want to talk to you a little bit about the digital resources that I generated using this specimen data coming mainly from the O'Brien collection. So I use Symbiota portals. I also use TaxonWorks and Taxon Pages. I share everything with GBIF. I use Bionomia to link the taxonomist and collectors data to the specimens. So everything starts just picking the specimen, identifying the specimen, and digitizing it directly into our Symbiota portal that is Ecdysis. So you can see all the information in this portal. You can go to ecdysis.org and the information will be there for every specimen. For very special specimens, or at least one for each genus or species, I also took a lot of images to illustrate the record. So we use Ecdysis to directly digitize the specimens and generate the information. But we also have a new portal that is called the Weevil portal that you can find in that URL, where we are looking to gather all the information that is available about weevils. So I have been aggregating, this is in collaboration with Jennifer Girón and Lindsay Walker from the Symbiota team. So we are trying to aggregate all the information produced. I have sent all the records digitized in Ecdysis. So we have a lot of points. The orange points that you can see there are from the ASU collections, plus collaborations with the Fossil Insect Collaboratives, ECN, and the Smithsonian and Harvard collections. We have also downloaded some records from GBIF, but I can say those are identified below or above the family level. So because we are using this new Symbiota portal, we can also play with the new functions. This is the new version of Symbiota. We can play a little bit with the maps. We can change the colors of the collections so we can see what we have already available in the portal. So we can also play with these very cool new heat maps. So we can check where we have records from. And for example, if anyone has specimens from El Salvador, I didn't find anything from there, from all the specimens that I checked or the specimens that are available online. So we are missing a lot of information also from Nicaragua, from a lot of regions of Guatemala as well. So besides the digitized specimen records that I share with this portal, I also have uploaded some images that I took during my visits to different museums or from the loans that I have received. So for example, I have images, even if they are not linked to a specific specimen, these specimens are still not digitized. They are at the Smithsonian. So I went there and really quickly took some photos with my phone, and I have those photos available. I uploaded them to the portal, into the portal, even if they are not completely digitized yet, but they are available there. So I also have a few images of part types and types. They are there currently available for everyone. So in this portal, we can also generate interactive checklists that have a lot of functions. You will hear a lot more about it during ECA, during Evan's presentation, so make sure you go. We can add a voucher. Jennifer also talked about this a little bit in the morning. So we can add images, we can add vouchers. But this, what I want to tell you, this is very interactive. We have all the new records, we have all the new information. Maybe some of those records are not still published. But I also wanted to generate these catalogs that are more static with the information that is already published. So it could be already available for everyone as well. So I am also using TaxonWorks to manage the taxonomic information. I am also uploading the images that are already available in the Symbiota portals. But we have maps with the information that is already published. So we don't have to, so we know that that's the actual current knowledge of the species, for example. I added all the images and all the names that are available, all the synonyms, for example, all the literature is already linked for most of the neotropical species. I still have to go through some of those groups, but most of that is already uploaded there. So just to finish, I want to share with you this very nice experience that I had during this work. A long time ago, in 2009, I participated in this fantastic project called the Llama Project. Probably many of you have worked with these specimens. And this little guy was collected there in Guatemala in 2009. I was part of the team that was collecting there. So eventually that specimen went to KU, to Kansas University, the University of Kansas, where it remained probably in ethanol for a few years. It was mounted and identified as Antribid, and it was digitized and it was there, it was available. So in 2022, I requested a loan and I got the specimen. I was able to identify it to species. That was a new record for Guatemala, was only reported for Belize. So that was very exciting. It's already published. And now this tiny Acarmimus sharpie has been updated in the Weevil portal with the image, with the ID, and it's also available as the species information is available in the Weevil portal. We have a few new records for Honduras. I haven't published that yet. I'm working on that. But in the taxon pages catalog, we have the already known distribution in Guatemala and Belize, the image, and all the information that is known about this species. So that's very cool. So whenever I return the loan very soon, I hope that the record will be updated in GBIF. But this, I just wanted to share with you that this is it can take a long time, from the specimen being collected to being curated, to being identified, to being published, to going back to the digital platforms with all the updates. So we have to be patient sometimes. So all the records are available in GBIF. And just to show you, with all this work in the O'Brien collection, we are now, or ASU has the higher number of anthribids in GBIF for now. Most of the material is neotropical. I wonder why. And the next collection is the museum, the National Museum in Costa Rica. They have a lot of neotropical material as well, but they have only identifications to family. So it will take for me to go there and identify the species at some point. Also, KU has a lot of neotropical material, and the Smithsonian has 14 neotropical specimens digitized. So I can do that at some point as well. So thank you very much. That is a very good question. I try to update the taxonomy as soon as I have changes, but with this group in the neotropics, there haven't been many changes in like what, 100 years. So it's it's a little easier to get the names, although the tribes, for example, have had a lot of changes and I have to keep up with that. Yeah, but as soon as I can, as soon as I see the changes, I will do it. Mhm. Thank you.
And next up, Oliver Keller.
Thanks. I invoked my presidential powers to get on the schedule. I'm not talking about an insect collection. So I just started a job at the University of Michigan in the new pathogen biorepository in January or in February. And I'm here to pitch collaborations. We are a repository. We have liquid nitrogen storage. So we are available if you have samples that you don't have storage capacity for. We have space. We have liquid nitrogen tanks. We have currently the capacity for about 100,000 samples. This is a collaboration between the University of Michigan Center for Infectious Disease Threats and the Ecology and Evolutionary Biology Department. And this is for pandemic prevention. So what I'm looking for is collaborations with you guys if you collect any insects or arthropods that are blood feeding or that are known to harbor any kind of pathogens that may be important for human health, right? And we will store them, we will digitize them, and we will make them available for loan to anyone that wants to work on them. We also work with mammals, so we have a lot of bat samples, mice samples, and stuff like tissue samples, but I'm in charge of growing the arthropod part of the collection. So I'm here to, see if I can collaborate with you guys. We will send you vials labeled with RNA DNA safe shield. So no costs for you. We pay for the shipping back to you and back. And then we will store the samples until they're used up in case someone actually does research with them. But what we're asking for is that you mainly collect live ones and pop them right into the RNA shield so we can facilitate RNA research. And if you want to collaborate, please use either of those two email addresses to contact us. The first one goes to my colleague, Nikde Ordonez, and my boss, Tim James, who is a fungi person. So we also do we would do any fungus specimens or so with pathogens. But yeah, please contact me. We can set something up. We can go the official way with that we set something up with the university, trying to avoid that because that's lawyers involved. All we would need is an email from you that you're donating the specimens to our collection, to transfer the rights to them, and then, but you will still get all the credits. You will be in the collection as the collector, where it came from and everything. But yeah, that's it. So yeah, if you want to talk, please find me. But if you want to collaborate, we'll send you all the material you need for this. Mosquitoes, ticks, I'm mainly interested in, but anything blood feeding, basically. So thank you. I guess I can take questions. There's enough time. Yes. Christy. Yes. Yes, there will be database, there will be push to GBIF, everything, like, yeah. I'm personally, yes, I have to clear it with my boss because of eventual space reasons, but I think, yeah, in the beginning, we were going to take all of that If there's maybe also like a economically importance to the pathogen, yeah, I would say yes for now. Yes, absolutely. Can you repeat that? I think for now it's more the New World. I think that's our main interest. So we also have a grant proposal in with the Belmont Forum in Europe to build liquid nitrogen facilities in 10 countries in the New World. So we have colleagues in Brazil, Peru, Guatemala, Honduras to provide them with liquid nitrogen storage to start building their own collection as well. So yeah, I'd say New World is preferred. We will take Old World samples, but New World is preferred. Thank you.